Okay, this is kind of where we left off. One of the predictions is that you'll find certain organisms with certain unique traits at certain places in the fossil record, because those traits evolved at certain times and certain taxa. So, for example, in terms of the vertebrates, there were these jawless fish. There was an extinct jawed fish called the placoderms. There were sharks and rays, and then bony fish, and then there was an ancestor of called a lungfish that was the ancestor of all tetrapods. And the evolution of four limbs obviously happened. You wouldn't expect it before things came onto land, right? In terms of the vertebrates. So you see it about the Devonian about 400 million years ago. The first vertebrates came onto the land. So in other words, if you actually, let's say, found a bunny rabbit fossil back in the Devonian, mammals didn't actually even begin to show up until about 220 million years ago. So then that would kind of blow up Darwin's idea of descent from a common ancestor. Okay, so let's, if we look up here, yeah, might as well get this going. Okay, good. If we actually look up here, we have a phylogeny. And what a phylogeny is that it shows evolutionary history. Time goes from older at the bottom to more recent up to the top. We see that backbones, vertebral columns, evolved first. In other words, a long time ago, even the jawless fishes, all vertebrates have those. Now then, in terms of tetrapods, they came later. Now it doesn't actually show relative rates of when it actually happened. But you can develop a phylogram that does that. This is called a cladogram. Okay? Now, and then you see four limbs, and then you see an amniotic egg. Amphibians don't have an amniotic egg. Birds and mammals, birds have feathers, mammals have fur and a mammary gland. So at the bottom here, it asks which trait first appeared in the fossil record in terms of that phylogram or cladogram. Huh? Backbones, exactly. And then, which taxa has a backbone? All of them, exactly. So, the most ancestral trait for this whole group is a backbone. These other traits define clades, that means define groups. So four limbs defines all the tetrapods, including the amphibians, the various reptiles, mammals, birds, all those things have definitive unique features that define them. So you would never expect to find things out of order. And if you did, like let's say footprints of dinosaurs around the time of when humans were around, well, that would be bad. Well, it wouldn't be bad, that would be the evidence. But you'd have to think long and hard about that. So, I don't think there were dinosaurs. We're long gone by the time of Adam and Eve, if there was ever an Adam and Eve. Okay. So here's homology again. All kinds. We went over this. You can see for the tetrapods, they use the same bones, but in different configurations, in the same place in the limbs, but the size of the bones, the shape of the bones have changed depending on how evolution has fashioned certain things. The same thing happens in terms of developmental history. You often see embryos kind of going through evolutionary history. So if you look on the bottom here, or in the middle, so this is a fish, this is a reptile, this is a bird, and this is a human, right? They all share the same developmental stages. They also show a tail and a gill slit. Even birds and humans and other reptiles who don't need gill slits, right? They don't get oxygen through gill slits, they get it through lungs. So if you actually look at embryos in evolutionary history, you can get a sense of how evolution proceeded. Same thing goes with the circulatory system. And then you often see the same genes modified in animals, even dissimilar animals. So we actually look up here. This is a gene involved in the eye, and you can see where there's been some substitutions in amino acids. And some of the same areas are substituted in humans who have aniridia, which is a disease where they can't see. And then this eyeless gene. Okay. Now, you can also see all these various individuals here, right? Different species. All great apes share a common ancestor right down here with squirrel monkeys. Okay? Now, no tail. There was also an increased flexibility of the thumb, more erect posture, and no tail, a larger brain. With the gorilla, reduced hairiness, elongated skull, all these changes happened, and you can find it in various species, just like we found with backbones and lungs and tetrapods and amniotic eggs. Delayed sexual maturity, increase in size of genital structures, but that varies depending on the mating system. So I mentioned that, let's see, chimpanzees have a mating system that's called promiscuous mating system. There's an alpha male, but he doesn't restrict access to the females. He just gets first dibs. Okay, so what there's going to be, since everybody else is going to also have sex with all the other males, or most of them, there's going to be what's called sperm competition. And so chimpanzees and bonobos have extremely large testicles compared to like us. So the more sperm they produce, the more likely that they're going to actually father the children. This is all part of sexual selection. So it's incorrect to say that humans evolved from monkeys. We shared along with the other great apes, the most recent common ancestor that was a monkey. We also, it's incorrect to say that we evolved from bacteria. We shared our most recent common ancestor with bacteria billions of years ago. So this is the kind of thing that you would expect to see. And there's another. Humans have 23 pairs of homologous chromosomes, as I mentioned earlier. We get one of the pairs from a mother, the other from a father. Well, it turns out the great apes have 24. So what happened to the chromosome? Anybody? Well, there's two possibilities. You could lose a chromosome, and or you could get fusion. Now, in terms of lost chromosome, why might this be unlikely? What do you think? Remember, chromosomes have all the genetic material, genes on them. Well, in fact, there was a major mutation where, but why would you actually, let's say, lose a whole chromosome? Why would it be unlikely? Think about, there's a lot of genes on it. What would happen to all those genes if you lost the whole chromosome? They'd be gone. Exactly. So you would probably end up, if you lost a whole chromosome or you had a lineage, you had a mutation like that, the fetus or the embryo would be lethal. It wouldn't actually make it. So it looks like actually what happened was there were two chromosomes that great apes have, 12 and 13, and they fused end to end. And you can actually look at the genome. And so one human chromosome should have homologous genes from two ape chromosomes. And this is exactly what we find out. Human chromosome 2 has the same genes as chromosomes 13 and 14 for apes. It also, as we'll learn, yes?
You just said 13 and 14, but up there it says 12 and 13.
Oh, sorry, my fault, 12 and 13.
Okay.
Okay, so, and it turns out homologous chromosomes in the middle, they have something called the centromere. And then at the ends of each chromosome, pairs, in each arm, they have what are called telomeres. Well, it turns out the telomere of human chromosome 2, the telomeres actually fused and created another centromere. So there's an extra centromere, like vestigial, and there's also two vestigial telomeres that wouldn't have been there, but we have this fusion event. So this supports the hypothesis that two ape chromosomes actually fused. Okay, homology. What's homology? Anybody?
Like something similar to something?
Exactly. Similar by descent from a common ancestor. So when we looked at the limbs of tetrapods, they all have the same bones, right? And it's because they all descended from an ancestor with those bones for the four limbs. Now, you can also get a situation where you don't descend from a common ancestor, where natural selection in different places but similar environments select for the same features. And that's called convergent evolution. So that's not descent from a common ancestor. It's actually evolution by natural selection. So, we think about flying. Insects fly. Birds fly. Bats fly. Pterosaurs used to fly. They're extinct now. Insects have actually lost flight, a number of lineages. So what happens here is that any lineage that for which flight might be useful, and you get, let's say you get mutations, and you ended up with a lineage that is can kind of lift off the ground. There might be selection for better and better wings. But the wings of an insect and the wings of a bat are not from descent from a common ancestor. They evolved independently, convergently. The same thing goes for the shape of a fish, whether it's a fish or a whale or a shark. They all look the same because a fusiform body enables an organism that lives in the water to move efficiently. So you see the same sorts of features over and over again. That's called convergent evolution. These different things exist primarily because the selective environment is similar, not because of descent from a common ancestor. So if you look on the left, most of those sandpipers have, a lot of them have short legs, but there was one change. Right there. And then that lineage split into two, but they both had, the ancestor for both, most recent ancestor, had long legs. That's descent from a common ancestor. On the right, long tails, all the other ones had short tails. It's most likely that at the very base, right here, the most recent common ancestor had short legs, right? And short tails. So what happened was, because of, let's say, sexual selection, females like long tails, something like that, or long tails are useful depending on where an animal lives. It looks like these things evolved independently or convergently, and not because of common ancestry. So remember, the ancestral trait was short tail, and these are now long-tailed, so they evolved independently. So one's the result of descent from a common ancestor, one is the result of evolution by natural selection for similar features because of similar selective environments. So, distinguishing homology from convergent evolution. We know what homology is. Similar features that are from descent from a common ancestor. In other words, the ancestor had those features, pass it on to all the descendants. Convergent evolution. That's evolution by natural selection of similar features because of similar selective pressures. If we classified dolphins and ichthyosaurs, because they look very similar, don't they? They both have a fusiform body plan, so they can move through the water. They're both top predators in the ocean. However, they developed independently. Dolphins actually evolved from a land animal that lived in a kind of an aquatic, semi-aquatic environment. And over time, you have this beautiful fossil record of the evolution of whales and dolphins. Came much later than ichthyosaurs. Okay? So now if you actually look down below, these are Hox genes that control development. Where, you know, the head, the limbs, the thorax, where everything is. It turns out that fruit fly Hox genes and human Hox complex genes, we share a lot of the same genes. The only difference is that they've been added on with vertebrates. So we have 13 Hox genes. Fruit flies have, 9, I guess. So you can actually see that this is convergent evolution. This is homology. We shared the same genes because we shared the same ancestors. It turns out the same genes are used over and over and over again instead of inventing new genes. Okay. Vestigial traits, we talked a little bit about this. As you know, I think I mentioned this earlier, that I've always felt like something in my life was lacking. I didn't know what it was. I went to shrinks about it. What's going on? Why do I feel like there's a part of me missing? What part do you think that is? The tail, exactly. It turns out that I miss my tail, right? And I mentioned how useful a tail would be, especially a prehensile tail, where you can grab onto things. So you probably might realize that some of the feelings that you have for you're missing something might be that. You might want to explore that with your shrink. Okay. Werewolf syndrome. I mentioned that. The other night, it was actually early in the morning, around 1, I saw so many coyotes. I saw coyotes around campus. I was driving up Lindley. I saw more coyotes. I saw coyotes in Encino. They were all over the place. It's just amazing. Anyway, so some people actually are born. Normally I get everybody to stand up and howl like wolf, but I'm going to maybe pass on that. Okay. Why are humans prone to choking? Well, there's a hypothesis here. If it was, yes.
They say that, well, from what I heard, that when you're falling asleep and you feel like you're falling, it's because it goes back to our genes to where you used to sleep on trees. We used to like catch ourselves from falling.
Oh.
Is that true?
I don't know. I don't know. It could be. Look it up. Go online and tell us, okay? Okay. So, if there was an intelligent designer, I don't think the intelligent designer would have designed things so that it makes humans more likely to choke than any other animals. Unless maybe you wanted a fail-safe mechanism to kill someone who was bad. You know, so instead of giving them a heart attack, you just give them, you know, you cause them to choke to death. But anyway, the intelligent design hypothesis is disproved. An intelligent designer would never actually look at why humans are prone to choking. This is actually the result of evolution. First, tetrapods evolved from a lungfish ancestor. Lungfish had lungs, but those lungs actually evolved from a swim bladder that most fish have to control where they are in the water column. Okay? Now, the lung developed from a swim bladder. A cross of the airway and GI tract developed into tetrapods. You wouldn't do that. You would actually have two separate openings, not through the mouth. And then what happens is for speech, and this evolved later, the larynx, which is where the vocal cords are, and the pharynx, it actually descends in an adult. And that descent allows us to make a whole bunch of other sounds. Unfortunately, it then causes the two GI tract and the trachea to be right next to each other instead of somewhat farther apart. And so we can speak, we actually are more prone to choking. Not very good design, but we get a benefit out of that, namely that we can speak. Of course, when you're choking, it's actually hard to say anything except, you know, kind of, and scream and, well, not muffled and turn red and then blue and then you die. Okay, so we see a lot of these things in evolutionary history of an organism, something that evolved from a common ancestor that had certain features, certain traits, to descendants that evolved new traits that might have from old traits. This is actually what's called the panda principle. If you actually look at a panda, they have an extra thumb. It's not from the same 5 digits that we have. They have 5 digits too. It's part of their paw. And in the bear ancestor, the paw was basically evolved to tear things apart and to run. Well, pandas don't necessarily do much of that. They eat bamboo all the time, and they need something to grab a hold of it. So this other bone, called the sesamoid bone, it over time became elongated and became what's an extra thumb, so it could grab hold of bamboo. This is called a panda principle. It works. It's not as good as one of our thumbs, but it works well enough. There's lots of transitional taxa. This example is a transition from our ancestors that were fish, a special kind of fish called the coelacanth or a lungfish, and then the first evolution of tetrapods. And you can see there were some new things that came along. The head is long and flat, the neck, fish don't have a neck. They can't look over their shoulder. They can't do that. The pectoral fins are weight-bearing with distinct jointed wrist and finger bones. Ribs are long and sturdy to support the whole body on land, which, in other words, now we've got gravity playing a major role, whereas when you're in the water, not so much. Small pelvic fins. And we've got this whole class of fossils that show the evolution from fish-like ancestor to tetrapods. We do the same thing with birds. Up on top is a bird-like tetrapod dinosaur. It couldn't fly. It had feathers. Feathers were used for, well, why do you think early feathers? What would be the use for early feathers? Anybody? What do you think?
Like warmth?
Yeah. Insulation. Anything else? Think about feathers now. Are they all the same color or not? Okay. Display. So we find out that the feathers on dinosaurs, they both had downy feathers for warmth, and they had feathers of different colors, and it looks like it was for display. And then Archaeopteryx, right below it, that's considered one of the first birds, or it could be a bird-like dinosaur. It shared features with dinosaurs, namely teeth and a tail that modern birds don't have. Birds have beaks, and they also lost their tail. So these are transitional taxa indicating descent from a common ancestor. So this is how we actually show evolutionary history in the terms of a phylogeny. you can actually, now we actually have different kinds of these phylogenies. Some actually just show, like this one, relative, doesn't show anything about time when things evolve. Traits are mapped onto it, amniotic egg, extensive changes in the skeleton, feathers, all that kind of stuff. But it shows an ancestor-descendant relationship. We also have some things called a phylogram that actually show time periods or how many changes occurred relative to other lineages. So this is a phylogenetic tree. The ancient lungfish is the outgroup, and limbs, and then all these other things that evolved specific to each group. As you notice, all groups from the amphibians all the way up to the top are tetrapods, so they all share 4 limbs. Even the ones, doesn't show here, like whales in mammals and snakes in squamoid reptiles. So how do we tell if a species are related? Well, they all are vertebral column, so if we use that, we wouldn't really be able to tell which came first. But it turns out there are other features that we can use to order things, and that is hair and mammary glands. Only the platypus, kangaroo, and beaver have hair and mammary glands. The turtle does not. So the turtle looks like, even though they all share backbones in a vertebral column, there's a new feature here that's evolved in mammals. Now, we can also, these are different kinds of mammals. A platypus is actually a mammal found in Australia that lays eggs. We can use egg laying, so probably the ancestor had laid eggs, and then eventually placental mammals developed. A placenta is used to nourish an offspring inside an individual. Okay, so it actually shows that kangaroos, which are marsupials, they're both placental animals, but marsupials are different. The very kind of, well, an embryo or fetus crawls out and then up into a pouch, latches onto a nipple, and then develops further in the pouch instead of staying inside like with mammals like beavers and ourselves. This is a dinosaur phylogeny. You can see where birds show up. They're actually part of tetrapod dinosaurs. There's a whole bunch of different kinds of dinosaurs. You can see how they actually split. There were a couple of major splits that then gave rise to other kinds of dinosaurs. Birds are more related to tetrapods like Tyrannosaurus rex than they are, let's say, to, what is that one? Right, Triceratops. This is the tree of life, same sort of thing. 3 domains, bacteria, archaea, and eukarya. Archaea and eukarya are actually more closely related. Eukarya is actually a combination. They actually have features of archaea and bacteria, both. Now, the Linnaean classification system, it was basically like this: domain, kingdom, phylum, class, order, family, genus, species. Now what this means is that the largest classification will be up at the top, domain or kingdom. And all these individuals, they're all animals. They all share common features with all animals. They're also all eukaryotes, obviously. However, the phylums are different. The two on the left and in the middle are part of the chordate phylum, and then the octopus is a mollusk. If we go down, let's say, to class Mammalia, that means they're mammals. Two of them are mammals. Cephalopod is a different class for the mollusks. And same thing with primates and cetacea. So as you get further down the list, you get more specific. You get to actual species. So we're Homo sapiens. The whale is Balanoptera musculus, the blue whale. So if we go from bottom to top, we go from more specific to less specific. So we talked about the forces of evolution, natural selection, evolution by natural selection. But there's the other forces too. There's mutation, genetic drift, non-random mating. Mutation is because of DNA replication that goes awry. There are 4 bases, as we'll learn later on. If you have a substitution for one, you're likely to change the amino acid. Amino acids make up proteins. So occasionally, random mutations are the sources, actually, of all new alleles. Most random mutations, if they're in a non-coding region, have very little effect. If they're in a coding region, they're usually negative, because they disrupt a protein that's configured very specifically. So if you change the amino acid, you're likely to change the protein in a way that makes it less functional. But occasionally you have mutations that are beneficial. In fact, you can even have, as we showed with chromosome 2 in humans, you can actually have mutations where whole chromosomes swap genes or fuse or things like that, or actually double. So instead of having a diploid organism, you have a tetraploid organism. So there's a reading frame. Each protein or each amino acid is composed of 3 letters. The 3 letters for DNA are adenine, thymine, cytosine, guanine. If you have a mutation where the reading frame flips, or you lose, let's say, one of the bases, you flip the whole reading frame, everything from that point on results in a mutation that gives you gibberish. In other words, it'll produce amino acids, but it'll produce a completely non-functional protein. Now, if you do the reading frame and let's say you lose 2, same thing. Because a codon is 3 letters instead of 2 or 1, if you shift the reading frame by dropping 3 letters, you'll actually end up back to producing a protein, but it might not be the exact one. So if you look at rat down there, if you remove that R, everything up to that R made an actual amino acid. And then at is where things went south. If you deleted 3, in other words, in a codon, if you had a 3 base pair deletion, you're still maintaining the reading frame, but you're going to likely have a different amino acid. So in this case, the fat cat ate the big rat, right? We dropped that. And now the fat cat ate the bat. still coding for something that's actually perhaps useful, a real amino acid, but it's a different one. Okay, genetic drift. We've talked about a little bit about it before. If you think about a gene pool with all the alleles in it, the chance, now there's genetic drift, I'll show you exactly why. Okay. The chance of you receiving your mother's father's chromosome or your mother's mother's chromosome is 50%, like flipping a coin. Got that? So genetic drift, I know it's anything random, like me stepping on the butterfly earlier that I explained, but in this case, in terms of producing gametes, you have a 50% chance of inheriting your mother's chromosome or your mother's father's chromosome, and on the paternal side, your father's mother or your father's father. Okay? It's like flipping a coin. Well, you can see how big of a variation there is if you only flip the coin 10 times. That would be like having a population size of 10. Okay? And so you end up with basically a 10% switch. 6 and 5, 6 out of 10 is 60%, 5 out of 10 is 50%. Occasionally you could even get two of the same versus the actual expected 5. Now, the thing is, if you go up and flip each, in other words, get which chromosome you get from each parent, let's say 100 times, that means a bigger population size, there's only a 1% difference between groups. Okay? So, in the case of group 1, let's say you flip it in one time, you flip 53 heads and 47 tails. Okay? Same thing in group 2, but it's 51 or 53 versus 54 is a lot less than 50 versus 60, which you get with a smaller population size. And then as you get up to a large population, 1,000, then it's 1 in 1,000. So this tells us something about how drift operates and where it might be strongest. Where might drift be strongest? In a small population or a large one?
Large.
No, small. A small population, you're likely for alleles to change, they can change by 10% versus 1% or 1 in 1,000, 1/10 of 1%. So actually, with a large population, it's likely that you won't be losing alleles or gaining, having alleles go to fixation, because it'll change much more slowly. Evolution actually works more efficiently with a large population than a small one. Drift is very strong in a small population. So, random fluctuations in allele frequencies from chance alone. In other words, do you get your mother's chromosome or your mother's father's chromosome? You can look on the left, population size of 4. This population, you can actually see either, actually go up a little bit, okay. Because it's such a small population size, in this case, this allele A1 actually went to fixation, and A2 went extinct. And if you had a whole bunch of different populations, it would be very random which alleles actually stayed in the population and which ones went extinct in the population, just from chance alone. When you actually have a large population size, let's say 400, drift is much weaker. And so the chances of losing an allele over many, many generations is reduced. The chances of an allele going to fixation is much reduced than with a large population. And in fact, natural selection can work much better with a large population. First of all, large populations usually have more alleles, more genetic variation to work with. So that's one thing. And then, of course, in large populations, drift is weak. So occasionally a population will go through what's called a genetic bottleneck. Turns out humans have gone through a number of genetic bottlenecks. All the humans that are today descended not from Africans, but left Africa, they went through a genetic bottleneck because only a small group actually migrated out of Africa. There were actually a bunch of different migrations of modern-like humans, but they didn't leave any descendants. When a small group starts a new population in a new place called the founder effect, we'll look at that. But it's a genetic bottleneck where you go from lots of genes, the founders now only have a subset of those genes that were there beforehand. So we see the same sort of thing. As you have a genetic bottleneck reduces the size of the population, so drift increases. Let's say high mortality or something like that. You're only left with a purple and blues instead of the reds. They're completely gone. And then the bottleneck population is likely to have different allele frequencies than the first one and be under strong drift. And it looks like that happened with elephant seals and cheetahs. There's also what's called the founder effect. Think about the Galapagos finches or the birds that populated Hawaii, the honey creepers. There was only one founding population. It was probably just a few birds, males and females, that got blown off track, say, during a hurricane and somehow landed on Hawaii intact and bred. That's a very small founding population. From that founding population, as new islands rose up and the other islands kind of got eroded away, you had birds flying from one to another and then developing into new species. Each of those times, though, there was small populations, small founder populations. So you go through successive bottlenecks where drift can be very, very strong. In this case, Tristan da Cunha, an island in the Atlantic, the British claimed the island. They colonized it with 15 British colonists in 1814. One of those individuals had a recessive allele that resulted in retinitis pigmentosa, makes you blind. 1 out of 15, now in the normal population, that allele would show up 1 out of 10,000 or 1 out of 100,000. So the chances of inheriting, a child inheriting it from both parents would be very low. But this is a small population, and it turns out this retinitis pigmentosa, even though it's a deleterious allele, it still drifted to high enough incidence that about, 10 times higher frequency on the island. So there's a lot of residents that either carry the gene that they can pass on, or they inherited both copies and they're blind. Non-random mating. You could, with a gene pool, and there's equal mating that's random mating. You can choose any allele just as likely as another allele in terms of mating. But a lot of animals don't mate randomly. They mate, let's say, assortatively. In other words, they choose someone like themselves, or maybe they like something novel and they choose a mate that's not like themselves. When you do this, you actually reduce the effective population size. In other words, you increase drift, and you lose genetic diversity, and you get something called inbreeding.
Like humans do?
Yeah, exactly. So, and there's very good examples of this, because you start seeing a higher incidence of these alleles that by themselves may not cause a problem, but two together, recessive, they do. So you look at the mummies or the Egyptians. Same thing with the royal families of Europe. They had incestuous marriages. They were more likely to marry cousins or even brothers and sisters, because the royal lineage was, of course, an example. They got their power descended from one of the gods. Well, you do that often enough, and you start seeing these recessive alleles happening. So like King Tut, he died very young. He was like 19. His parents were a brother and sister. He ended up with a club foot, necrotic dead foot bones, a cleft palate. His immune system was not as good as someone else, because if you end up with more diverse immune alleles, you're better off fighting infections. He also ended up with the father of 2 stillborn fetuses, which isn't at all uncommon. The same thing happened in Europe. There were a lot of hemophilia is an autosomal recessive trait. It turns out it's a sex-linked trait. It's on the X chromosome. So if you're a female and you inherit the trait, you don't get it. You're just a carrier. But males don't have 2 X's. Females have an X and another X, and it's called a rescue effect. For males, it's not like that. They have a Y. So essentially there's no rescue effect, and they actually show. So you get hemophilia. Color blindness is only found in males unless a female is actually unlucky enough to inherit 2 copies, 2 bad copies, and it's very unlikely. And then there's migration. Migration actually homogenizes populations. It can be migration or dispersal, whether it's animals moving, whether it's the seeds of plants moving, bacteria on things that are moved by animals, let's say like that. Pollen. So it's the movement of alleles from one population to another. Like I mentioned, what it does is it increases gene flow between the populations, and you start to see that one population is homozygous A2, the other is homozygous A1. Over time, they start sharing the same alleles, depending on how much migration. When gene flow is interrupted, populations are then on their own evolutionary trajectory, and they're likely to change, whether from drift or from natural selection. So if you've ever been to the Grand Canyon, there's these squirrels, and on the south side, this is it's the ancient squirrel. before there was a big deep canyon here. On the north side, there's the castle-eared squirrel. They're related, but they were split apart once the Colorado River started carving the canyon a few million years ago, and they've now evolved independently. So when you get migration, you're less likely to have independent evolution. When you interrupt gene flow, population differentiation, genetic divergence, you can actually result in what's called speciation, which we're going to get to shortly, like right now. So the basis of creating new species is often geographical because of some barrier to gene flow. Okay, gene pools, the size matters. As I mentioned earlier, drift is stronger in small populations with small gene pools. If you look here, if you look at a small gene pool, that's a smaller gene pool. That's a smaller gene pool. This is part of a larger, somewhat larger gene pool, but smaller. And then the whole gene pool. Okay? In the whole gene pool, there are alleles that are not found in the smaller gene pools. So natural selection can actually, in a large gene pool, can actually work more efficiently because it has more variation to work with. So why are, in the face of environmental change, new selective pressure, which is more likely to go extinct? So species with a small or a large gene pool.
Small.
Right. Why are they more likely to go extinct? They have fewer alleles. Genetic drift also has a larger effect. And so both A and B are true. Species are more likely to evolve rapidly by natural selection if they have a small population and a small gene pool, or a large population and a large gene pool. Large. Evolution is much more efficient with more alleles to choose from than a small population. Okay, speciation. Where do species actually come from? First of all, the first thing we should probably do is, what's a species? Giraffe is a species. It's kind of like, I know one when I see one. It's kind of like pornography, right? So what's a species? Anybody? What do you think back there? How would you define a species? Don't worry about everybody else.
I don't know how to say it without saying
Okay. They have different characteristics. Species have very different characteristics from each other. How's that? Oops. Okay. Anything else? Are species more likely to breed with each other? Being able to breed with different species more likely to breed with each other successfully or not successfully? Huh? Not. Okay. They share different genes, and the genes actually don't line up. They share different chromosomes number. So okay. So, we'll say that species are the smallest evolutionarily independent unit, but that's pretty arbitrary. Evolutionary independence, like those squirrels on the north and south rim of the Grand Canyon, that implies independent mutation, selection. They're all off on their own evolutionary trajectory. They don't share alleles anymore. Gene flow and drift operate on populations separately. Selection, mutation, and selection, all the forces. Species consist of interbreeding populations that evolve independently of other populations. In other words, they usually don't breed successfully. The thing is, there's a whole bunch of different definitions of species. So species are kind of like arbitrary, but they're not arbitrary like classes like small, medium, and large. Those are real arbitrary classes. Species, they're dynamic, changes in allele frequencies over time, so there is kind of a fuzzy boundary around them. They're not exactly completely arbitrary, but they're not like a class like small, medium, and large. They actually have something to do with biology here. So, it turns out you can have a species like Canis familiaris, or dogs. They've been bred for different things, and you can have incredible differences in anatomy and physiology. These two animals can still breed together. Hopefully, it would be the one on the right is a female versus the one on the left. Okay? Of course, the one on the left would have to be helped, picked up and, right, allowed to mate. Okay, now, leopards and lions that are both Panthera, from the same genus, and they share a lot of alleles together, but they're separate species. So looks alone are not enough. So here are some definitions. There's a morphospecies concept, anatomy. So fossils, you don't fossilize behavior, you don't fossilize physiology. Usually you fossilize hard things. So the only thing you can actually use are morphological traits, anatomical traits. And then you can maybe see through the fossil record how certain lineages change over time. There's problems in that you really don't know very much about how related two species are without, let's say, DNA and some other features. So it's kind of like, I know one when I see one. The biological species concept is the one that's most often used, but it's not entirely workable. It's a group or population of actually or potentially interbreeding organisms that can mate and produce fertile offspring. Well, we don't know in terms of fossils. We can't use the biological species concept because we don't know how they mated and who they mated with. Some organisms reproduce asexually. Okay, and then they also, you can actually end up with hybrids, either that are fertile or not fertile, but especially when they're fertile, those hybrids, they might have lower fitness, but not necessarily. And so it kind of breaks down with hybrids. And then there's a phylogenetic species concept. The smallest set of organisms that share an ancestor and can be distinguished from other such sets. The primary problem is populations could be separate species. Like how far down do you go? The biological species concept is a mechanism. The phylogenetic species concept is actually a pattern. One looks back in time versus forward in time. So, like I said, the biological species concept is most often used, but you really have to, if you're doing a study on, let's say, hybridization and things like that, you have to define what species concept you're actually using. So, are you in my species? I'd rather not find out. Okay. Now, speciation is actually a three-step process. Usually it involves a geographic barrier. So you have a population that's homogeneous in one area, and a barrier somehow splits the population into 2 parts or 3 parts or whatever. Those populations no longer share the same alleles over time. They have different mutations, different alleles develop. Selection works, perhaps the different areas are different in terms of the selective environment. Genetic drift perhaps is stronger now because you initially had a big population, now you have a couple of smaller populations. Eventually, over time, populations accumulate differences in alleles from all these different forces. And then eventually you end up with what's called reproductive isolation. If they were brought back together, they can no longer breed successfully, or they won't breed. So this is kind of a figure that kind of shows what happens. Right here, come on. That's one whole population in the past. Eventually, there's a barrier, let's say like the Colorado River and the Grand Canyon. All these different lineages are lineages of alleles. Some alleles end up in one group more likely than another, and then once they split, they also are now on different evolutionary trajectories because of mutation, drift, random mating, and natural selection. Eventually, they develop so that they're actually separate species. So there's some modes of speciation. The most common one is the one where we have 2 separate locations. That's called allopatric speciation. And it's usually by vicariance. Vicariants are geological formations in general that split species, whether it's a mountain rising up, a river splitting populations in 2, things like that. There's also what's called another kind of vicariance, or parapatric, but it's actually not vicariance, it's actually by dispersal, like founder effect. You have a population on a mainland, and some individuals actually fly away and land on an island. That creates a founder effect. There are some instances where they're next to each other. There's even speciation where you end up with in the same area. Okay, so this is what it looks like. We have this homogeneous population. River splits it. If you look in the upper right-hand corner, left bank, right bank, you have, after 20 generations, some differences have evolved in terms of alleles, more differences. Some have evolved spots. And then finally, even more differences after 60 generations. So eventually, these species might not even recognize each other for breeding if let's say the river dried up. This is another example of allopatry. The Panama Canal, or the Central America, the Isthmus, that developed about 3 million years ago. Before that, there was the same species of these snapping shrimp. They were one homogeneous population. As the land rose up, and also the water levels dropped, you ended up having separate species, or they might be related. You might find, let's say, this one and this one are more related to each other than this one and this one, because they share a more recent ancestor than these other species. Okay? So, now, of course, with the Panama Canal and maybe a new canal being built, there's now some of those old species are coming back together. Parapatric. So you have a mainland population, a small island. There's a bridge. Some get on the island, some are on the mainland. The ones on the island are mostly kind of darker green and light green. And then the ones on the mainland, the same sort of thing, but they're going to start changing. Spotted mutation ends up. And so eventually they might develop into new species. Dispersal, like this is what happened with Hawaii. There were a whole bunch of organisms that colonized Hawaii. No mammals, but there were birds. There were Drosophila, which are fruit flies, and there were also a lot of plants. And they speciated rapidly. And then sympatric speciation is where we're going to end up. You start out with a population, and you have different alleles. New populations, they might be feeding on something different because of, let's say, in fact, I'll show you. You have these moths, apple flies and hawthorn flies. Originally, there were no apples in the United States. It was just hawthorns. They're attracted to hawthorn trees. They can eat hawthorn leaves without getting killed by the toxin. And so they specialize on hawthorn trees. When apples came along, some of them actually started breeding and doing their whole life cycle on apples. Over time, the populations of flies started to diverge. Even though they were in the same place, there were little pockets, in other words, microhabitats like apples or hawthorn, where they were more adapted. So this is sympatric speciation in the same place. Allopatric speciation, different places, whether it's from vicariance or from moving from one place to another. Okay. We'll call it there, and we'll talk later about a little bit further, and we'll finish up.